Thank you for a nice introduction and also inviting me again to this amazing international symposium. And today I'm going to talk about our recent approach to build a cross-tissue single-cell and spatial atlas across human diseases. So let me start with the introduction about what our future aim is. So our ultimate goal is to understand our body as a multi-scale gene regulatory network. And by multi-scale, I usually think about three layers. The first layer is the intracellular gene regulatory network, which we deal about how this pluripotent stem cell differentiated to each of the fully differentiated cells and how the transcription factors are regulating each path. And then we can think about intercellular gene regulatory network, where we can imagine there are two cell types A and B, and how each of the gene I in cell A is quantitatively affecting the gene function of gene J in cell B. There can be so many combinations between different cell types. So we are, I think, actively unraveling this intercellular gene regulatory network. And after that, we would have the tissue map, tissue atlas, or tissue predictive map for such as organ like a lung and liver. And after that, we would need to build a multi-organ gene regulatory network, thinking about how the gene program in one tissue can affect the program in other tissue. And eventually, we would like to achieve this human digital twin, the fully full computational copy of the biological entity of a human in computer. And we would like to have it as a predictive power across lifetime and across many diseases. So today I'm going to talk about what kind of bioinformatic framework would we need to achieve this goal. And for this, of course, the single-cell technology has been really good at unraveling the intracellular gene regulatory network, how the cells, the intracellular gene regulatory network is guiding the differentiation path of each cell. And then eventually, nowadays, there are so many spatially resolved transcriptomics that is also advancing technologically, and also a lot of data is coming out, which is giving us information about which cell is sitting next to each other and how they are interacting with each other physically. And this now allows us to build a good intercellular gene regulatory network. And today I want to start by showing you one example, our recent example on pancreatic cancer that we both applied single cell technologies and spatial technologies to understand the cancer evolution. So if you look at the UMAP on the left, this is the batch integrated UMAP from about 20 different patients, the pancreatic cancer patients. And what you can see is there are normal cells, normal epithelial cells, and there are epithelial cell type that represents the pre-malignant status. And then there's a classical type of the cancer cell and basal type of the cancer cell. And we can see there's a transitioning intermediate type of the cancer cell. And usually in pancreatic cancer, this basal type is known to have the bad prognosis. And we have postulated that maybe the evolutionary path of the cancer is starting from this normal to pre-malignant status, to the classical, to the transitioning, to more basal type, because this is what we can know from the transcriptional similarity in UMAP. And then what we could also see is the fibroblasts, which do not have any mutation, also shows a very interesting change upon this malignant transformation. For example, in pancreas, usually you have this Stillay cell as a normal stroma, but then in pre-malignant status, we can see very recurring status of the pre-malignant specific fibroblast cell type, which express COL9A1 as a marker gene. And after that, as the cancer becomes malignant, we can see the diversification of this cancer-associated fibroblasts into three phenotypes. One is ECM depositing COF, expressing LRRC15 as a marker gene. The other is ICOF, usually immune recruiting or inflammatory, expressing SFRP1 as a marker gene. And then we found kind of novel COF type, which expresses STRA6 as a marker gene. But we were not sure about what the function of this cell type was. And then after that, we selected about 8 patients from the patients that we profiled as single cell. And then we performed the visual analysis, the spatial transcriptomics, which gives you kind of low resolution spatial map of the tissue. So each of the spot in the vision would represent usually 5 or 50 cells in a single spot. So what we need to do is for each spot, we need to utilize our single cell profile to deconvolute this spot. And then we color them based on the suggested or predicted abundance of this single cell profile on this Spatium data. And what was very evident from this data was that, as you can see in this patient's spatial data, there is a gradient of the cells starting from the orange or brownish color to the green to the purple. And what it matches to is this orange color matches to the classical type of the single cell. And then this green color matches to the transitioning type of the single cell. And this purple color matches to the basal type of the single cancer cells. This kind of shows that the predicted evolutionary path from the single cell transcriptome data is also visible in the cancer spatial data. So it's almost like a sedimentary rocks that shows the dynamics and evolutionary path of the cancer. And in addition, what you can know from here is that this yellow color represents this ECM cuff, and they are all dispersed throughout the cancer bed, right? So these three colors are all cancers, and these yellow colors are overlapped completely with these cancers. So this ECM cuff is co-localizing with all these cancer cells, whereas this I cuff, which is colored as blue here, you can see that they are completely isolated from the cancer bed, and they occupy quite far distance from the cancer area. And if we kind of summarize this with this kind of neighborhood plot, which we depict each node here is single cell type, and the edges connects the cell types that are closely co-localized within the spatial data. And as you can see, the putative cancer progression axis is really visible in the spatial data. And this LRS15 fibroblast, the ECM fibroblast is, as I show you, in many examples, they co-localize with the cancer in pancreatic cancer sample. And then they also co-localize with the macrophages. So this represents the epithelial niche enriched with LRS15 fibroblast. And then this ICAF, SFRP1 fibroblast, localized in immune niche, they co-localize with endothelial cells and T cells and dendritic cells and B cells, but then they do not meet with the epithelial cells here. So I usually call this the South Korea and North Korea plot, where they are not really mixing with each other. And so for the efficient cancer immunotherapy, maybe we need to find somehow the way for these immune cells to enter the cancer area. And then the next question is that, is this generalizable to other cancers, this spatial relationship? Or if you look into other cancer data set, do we see a mixture of the variety of the spatial niche patterns? So that's the reason why we got interested in the big data integration. And obviously, as you can know, both single cell data and spatial data, they are continuously being deposited in public repository. And these numbers, I have collected them a few years ago. So now this must have increased a lot. And this data now encompass a lot of organ, developmental stage, and disease status. So now our actual challenge is how to integrate them into the single entity. And my previous background was to developing an algorithm for the efficient large-scale single-cell data integration. So I was originally really interested in this single-cell data integration task. And also I knew that when you aim to do the big data integration, that you need to start from some small toy example that you can play with. So I choose this tumor heterogeneity problem as my toy example to start with, because the tumors have enormous intra-tumor heterogeneity. So all the cell types that you can imagine can be represented in each tumor tissue. And again, the tumor can arise from the multiple organs. So we can also see the intra-tumor heterogeneity. And by comparing the tumor in microenvironment within organ and across multiple organs, then we can basically handle the task of the multi-organ data integration. So our original first question was, can we understand the generality and specificity of the tumor microenvironment? So for that, we have made this large-scale single-cell data collection. So for this data collection, we focused on the single platform, the 10x genomics platform, to minimize the technical batch effect. And we only focus on the unsorted samples because we want to understand the cell-cell interactions. So we need a sample that represents all the cells evenly. So we did a manual collection of the single-cell data across the tumor sample and normal sample across more than 30 different organs. So now this has featured about 1,000 individuals across multiple tumor type and about 5 individuals for the normal tissue type. And from this analysis, we can do the batch integration and then put all these cells into the same single UMAP space. And this is how the UMAP space looks like. And you can see that multi-organ, and normal tumor data is all intermingled in here. And from this data, we can annotate each cells with our own way so that we have our own axis that is uniformly applied to all different data. Then we can ask this kind of question. Can we define the hallmark of the tumor microenvironment? For example, CD8 T cells, NK cells, Tregs, and macrophage, and fibroblast, which exist in all tumor microenvironment. What are the genes that are commonly upregulated or downregulated? And you can see there are many interesting examples of the tumor-specific gene expression pattern across multiple cell types. And after that, while doing this, we realized that actually we can turn this effort to find the markers that can target the cancer cells for CAR-T or ADC therapy. So we took our cancer atlas and normal cell atlas, and then we applied the machine learning algorithm to find the combinatorial markers that specify the cancer cell against all other normal cells. And the reason why we focus on the combinatorial markers, there are two regions. One is the practical region. Nowadays, the CAR-T and ADC, antibody-dependent cytotoxicity mechanism, they actually have the modalities that we can logically and practically make this combinatorial targeting scheme applicable to the cancer. So we thought that there was a practical need for this combinatorial marker finding. The next thing is when we try to find the marker, the unique marker that specify cancer against all other normal cells, we realized that there's no single marker really that is specific to the cancer. So we come up with the AND or not logic gate markers, combinatorial markers that specifically target cancer cells, so have a high coverage on cancer cells, whereas they have a minimal coverage on any other normal cells in normal cell atlas. So compared to the previously existing CAR-T targets, we could see that this combinatorial approach can really increase the specificity while we can still predict the potential off-target for those small coverage on normal cells. What kind of cell types in normal tissue can be really targeted by the suggested marker combination that we made. And another thing that we did in this analysis, if you look at this UMAP, you will realize that although this has been batch integrated, the resolution might not look ideal. And one reason for that is at the moment, we did not have enough time to go through all the FastQ files and reprocess everything, remap them. So we start from the count metrics from each data. And this count matrix already contains a lot of bioinformatical batch effect. So to circumvent this problem, we had developed this NF-based cell projection approach, which I will explain you with this diagram. So in here, what we have done is we started from the roughly annotated large cell types. And of these cell types, we took each batch samples, and then we ran NMF with the running K parameters on each batch for each cell type, which results in a lot of NMF vectors, right? So to harmonize those NMF vectors, we projected those NMF vectors onto the UMAP space, and then did the same approach that we would do for the single cell on these NMF vectors, and then turn them into the NMF programs, the cluster of the NMF vectors that are recurrently found across batches within certain cell type. So for example, this is the UMAP of the mesenchymal NMF programs, which we have clustered and annotated. And then we took this, and then from these vectors, we have identified the orthogonal sort of marker genes. And using those marker genes as the reference component for the projection, we reprojected our cells onto the UMAP space. So this is now the NMF vector space, and this is now cell space. And you can see that the cell resolution has become much, much greater when you compare to the fuzzy resolution that we originally had. So we took this NMF clustering approach for a lot of different type of analysis. And from this, we could see that which programs are really truly niched in cancer, for example. So these are finding the true cancer-associated fibroblast based on measuring the ROE of the mesenchymal cell programs compared to the tumor and normal. So we can identify these three are truly cup that is usually not found in the normal cell. And also we can see whether there's any organ-specific bias in the abundance of these cell programs. And we could also validate them using the immunofluorescence approaches. And from this study, that I just want to highlight one thing, that we also did a co-occurrence analysis, which is that because now we have 1,000 tumor sample and 500 normal sample, and we have only collected the unsorted data, we can ask which multicellular programs are co-occurring in certain samples. And this is the network plot based on this co-occurrence analysis. So for example, this red circled module here, this is the multicellular programs. They only shows the co-occurrence in tumor tissue, and they are quite dispersed in normal tissue. So these are the tumor-specific co-occurring program, but they are not found in all tumors. They are found in only some of the tumors. And if you look at this multicellular program, then we can know that there are a lot of factors that defines the hot immune environment in the tumor, especially the components that constitutes the tertiary lymphoid structure in the tumor. And because this tertiary lymphoid structure has recently been suggested as a predictive marker for the response to the immunotherapy, so what we did is we took a lot of bulk analytic data, which has the response information to the immunotherapy, and then we deconvoluted them with our single-cell NMF programs. And what we found is all the factors that are co-occurring and forming a module here in this co-occurrence analysis, they have a predictive power on the response to the immunotherapy. So they come together. This multicellular program comprises the hot immune community that predicts and that sort of acts as a predicting factor to the response when the patients are given immunotherapy. So this is a summary of our toy example. If you remember, I told you that I thought that the tumor microenvironment sort of integration projects can be considered as a toy example. And now we have successfully finished our sort of toy analysis. So after that, we want to move on to our actual aim, trying to make the unified theory for our immune system across all diverse contexts, because the immune system has a yin and yang. So cancer immunity would represent the yin immunity, which is the repressive type, the tolerogenic immunity, whereas the yang immunity represents the highly active autoimmune or inflammatory type of the immunity. And for that, what we need is we need to collect the data set from more context. And actually what we have been doing was like this, the gold panning. So people would search through the mud stream and try to find the bit of the gold that gives us some of the useful information. But then it is highly labor intensive and it is not scalable. So we cannot scale this approach with this kind of the gold panning approach to the multiple context. But at the same time, I saw this nice image from the Google that showing that there's actually automatic gold panner. So you can make a machine which does this gold collection job instead of the human hands. So we thought that maybe then we can develop automatic gold panner for the single cell data. So we come up with this web crawling based public data collection approach, where our web crawler is continuously searching for the FASTQ files in GEO database. And then whenever it catches the new FASTQ file, then it would automatically download the data. Then our students has implemented a robust QC metric to get rid of all the low QC data. And with all the high QC data, we have classified them into the in vitro culture sample and fetal sample and all other primary human in vivo data, which can be also further subdivided into the cancer and non-cancer patient samples. And this is our realigned data set. So we are starting from the FASTQ and we are realigning everything with our bioinformatic pipeline. So they do not have the bioinformatic batch effect. But some of the big data that is not available as FASTQ files, we also generated the second tier data set that we only have curated their metadata with the same logic that we do for the first tier data set. And actually this features much more cells because it's easier to collect. But because the metadata is coherently annotated, we can utilize this first tier data set as a discovery cohort, and we can use this curated data set as a validation cohort. And this infographic shows what kind of data sets are collected in our database. So bloods are the majority, but we also have a brain, bone marrow, intestine, and liver, multi-organ sample collecting, coming from the, for example, COVID-19 or diabetes, Alzheimer or IBD, and many other types of the disease, including cancers. And because we have remapped everything, now I can say that our cell annotation here is quite robust and confident. So for the majority of the cell types that can be found across multiple organs, we sub-cluster them and then do the sub-cell type annotation with the highest resolution. So we have now about 165 cell types that has annotated on the immune cells and mesenchymal cells and epithelial cells. And brain cells, especially neuron cells, we are developing another version of the high resolution cell annotation, which also contains more than 100 or 200 cell types. So now we have about 300 cell types that has been identified across tissue. But now, because these cells are coming from multiple contexts, more than 100 tissue disease contexts, so now we can say that the cell type annotation that we have made represents most of the, not all, but most of the human virus that we can expect from the adult humans. And this cell annotation system, when we compare with the existing large-scale atlases, we are seeing that we kind of get the right cells, the common cells as a correct one, and also we have additional resolution, usually in immune cells and fibroblasts. And this is one benchmarking. We are doing a lot of benchmarkings. This is the benchmarking on the lung cell atlas, which has been published last year. And this is the original annotation. And this is the systematic annotation that has been derived from our model. We are calling the Pangea. There are level 1 and level 2 annotation system. And with the level 2, we can have this much of the resolution. And then if we compare each of them by drawing the matching matrix, what we can see is that for the epithelial cells, this long atlas, the organ-specific atlas, has a better resolution. But for the other universal cells, our atlas has a much better resolution. So it's kind of compensatory to each other. And this Pangea annotation provides the universal view of each data set. So we can apply this Pangea annotation to lung, liver, and intestine, and many other organs. And then you get the exactly same view of the cells from the same annotation. So that's what we are trying to achieve here. The universal annotation that can be found, that can be used in any tissue to guide you through the single cell data set. And from this data collection, because now we have a lot of ages that has been collected from our data set, we can see what kind of cells, for example, in blood are showing the correlation with the age. So whether they are proportionally increasing or decreasing across aging. And using those markers, we can also predict the new blood sample for their chronological age. And usually, when we make this regression model using the T cells and NK cells, the age prediction score is really high. And B cell and monocytes, they do have some age prediction power, but some other types, like dendritic cells, they do not usually correlate with the age-associated changes. And also, what we could achieve from here is that we can, now we have a universal cell type annotation system across multiple contexts, so we can ask their context specificity. So here we are classifying the cells based on whether they are organ-specific or they are disease-specific or they are healthy or universal. So these ones, like 81 cells, they are lung-specific. So these ones are usually epithelial cells or some organ-specific function cells. But this one, on the right panel, these are the ones that are universally found across organ, but that are also found in multiple types of the diseases. So these are pathology-associated cell types that might be interested in many medical researchers. And using these cell types, now we can do the blood phenotyping based on their abundance profiles. So we are calculating the abundance proportion of these cells that are found across multiple organs, but they are also disease-specific. And then now this clustering result, these dots here are not single cell, they are single sample. They are one sample of the single cell data that has been clustered based on our view of the cells. And we call this blood phenotype, the abundance phenotype of the blood samples. And there are about roughly 10 blood phenotypes that we could identify from the initial draft of the analysis. And then we can see that some blood phenotypes are enriched with the disease context samples, and some blood phenotypes are enriched with the control healthy samples. Then can we actually see multiple diseases converging on this same blood phenotype? The answer is yes. For example, in this BP7, we can see SLE and rheumatoid arthritis is converging on this blood phenotype. And there are specific cell types that are sort of resulting in this kind of blood phenotype. And BP4 highlights has the Alzheimer and neuroinflammatory disease in it. And then it has a high score of the CD8 TEMNA cells. So this is also converging feature of the neuroinflammatory or neurodegenerative disease. And this BP9 is, they contains COVID-19 samples from multiple different data sets, and they shows the multi-cell type strong interferon response as their characteristic. And if you look at this kind of blood phenotype in detail, for example, the monocyte interferon program is found both in BP7, the rheumatoid program, and BP9, the interferon program based on the COVID-19. But then the B cell and T cell interferon response is only visible in the COVID-19 interferon program. And if you look at individual COVID-19 datasets, then you can see that this BP9 phenotype emerge at really early stage of the infection, and then they go away quickly during the cycle of the infection. And next, what we are aiming to do more ambitiously is then can we actually also cluster tissue samples based on these cell proportions? So for this purpose, we focused on the cells that are shared in multiple organs. For example, macrophage, fibroblast, endothelial cells, and mural cells, and T cells and NK cells. As you can see here, that these combinations are usually found in most of the organ data that we have collected. So now we can use these 5 cells as a reference and their subcell type proportion as a feature to do the tissue sample clustering. And this map is now showing the tissue sample abundance phenotype. So each dot here is a single tissue data coming from each organ and each disease context. And as you can see here, again, there are disease-enriched tissue phenotype and tissue phenotype enriched with the control samples. And if we zoom into this profile, the detail, then we can see that one tissue phenotype, TP4 as we named, it has a converging nature coming from a lot of cancer specimens and fibrotic disease specimens. And there are clear 16 cell types that identify this cancer program that is recurrently found in multiple organs. And then there's another cluster, TP9, where you can see that the immune disorder disease samples are converging onto this TP9 cluster, which you can see from here, TP9. And so this shows again already that there are yin and yang of the immune system well represented in our data. So if we do the co-occurrence analysis again with these cells that has annotated across multiple contexts, then you can see that which cells are co-working together or co-occurring together. And these programs represents the immune activation related cells across multiple cell types. And then there's a hypoxic response, and related to that, you can see the exhaustion program coming in. And it is somewhat related with the interferon response, but it shows the lowest correlation with the fibroblast-based interferon response. So there must be also some dynamics in interferon response that is leading to the eventual exhaustion in cancer. And then you can see that it is also showing some interaction with the tissue damage responses. So this is already now revealing some interesting dynamics that is governing the tissue biology. And finally, what I want to highlight that as a use case of our data is that we can actually now do the spatial data deconversion using the universal cell atlas. So in any spatial platform, you would actually need a single cell reference to do the single cell resolution analysis. But now, because we have built a representative cell atlas across multiple organ and multiple disease type, so we can utilize this to deconvolute all the spatial transcriptome data that we have collected. And this is the sanity check result specific organ-specific cell type that we have identified in our single-cell study is also shown to be organ-specific in the spatial transcriptome deconvolution approaches. And one example here, this is the brain tumor data that we have reanalyzed. And you can see that there are paratumor area and tumor core area here in this brain tumor. And paratumor area has been mapped as neurons, which makes sense. And this tumor core, we can find all those tumor I forgot, TP4 program, which is cancer specific, is indeed niche in this tumor core. And this can be found, confirmed in multiple different types of the tumors. Or this single cell defined tumor program also shows the tumor specific distribution pattern in spatial data. And then we can also make the co-localization map using this 300 spatial data that has been universally deconvoluted. And for example, we start to see very interesting relationship between unknown relationship between cell types. For example, this CXCR4 positive endothelial cells shows the clear overlap with the TNF-alpha-6 expressing mural cells. So there are some co-localizing and maybe potentially also cooperation between the specific endothelial and neural cell type. And then going back to the original pancreatic cancer study that I opened my talk, this is the South Korea and North Korea diagram, which we show that the LRS15, the fibroblast, is interacting with all the epithelial cells in pancreatic cancer. And when we reanalyze with our universal cell annotation system with our own pancreatic cancer spatial data, the published data, then we can see that this epithelial cancer bed is co-localizing with LRS15. This makes sense. But then when we look at other cancer data that we have reanalyzed, interestingly, while this LRS15 fibroblast is really cancer specific, it belongs to TP4 program, but they do not overlap often with the epithelial cells. Actually, you can see that they are kind of localized in the boundary of the cancer instead of going into the inside of the epithelial core. And because we have now deconvoluted multiple cancer spatial data set, we can actually measure the distance from the epithelial core for this LRS15 fibroblast. And what was interesting was that it was really only the pancreatic cancer that we can see the LRS15 is niched in the epithelial core. And other cancers, they have a different sort of grade, but usually this LRS15 fibroblast is excluded from the epithelial core. And then what we can also see is it shows a negative correlation with the abundance of this exhausted CD8 T cell at the epithelial core. And it also shows a negative correlation with the average objective response rate to the immunotherapy. So now what we are thinking is that not just the abundance of the LRS15 fibroblast, but actually it's the spatial pattern. The tumor core infiltration of the LRS15 fibroblast is explaining whether the patients can be responding or non-responding to the immunotherapy. So that was one main take-home message from my talk today. And finally, I would like to end my talk by just sharing you with some interesting perspective that I think, and the perspective starts from our manual effort. So here, this is one of the metadata that we are automatically collecting the FASTQ files, but then we are not automatically collecting the metadata. For each of the metadata, we actually need to go through the GEO web page and also go through the paper and maybe sometimes into the supplementary table to turn all this GEO ID that we have collected to this nicely organized metadata table. And this is still a manual effort. It took 4 students' effort, about 4 months, to cover 4,000 samples that I show you today. So this is, again, not scalable. And it's not, considering the rate of the single cell and spatial data publication, I think no manual effort can now catch up with this speed. But then what we thought was that we can maybe utilize LLM as if they were doing the job that they can replace the job that our students has been doing for the manual metadata curation, because that's what LLM is about. So we collaborate with Professor Bo Wang in Vector Institute, and then we have developed this pipeline that is mimicking our manual approach. We are doing prompt engineering to let ChatGPT-4 to ask this question instead of us. And the accuracy score that we get for multiple categories range from about 85% or 95%. So there is a hallucination. Some of things are actually missing from the GEO. So we need to go to the supplementary table, which we are not being able to fit into the LLM models at the moment. And some, they just GPT make mistakes. So we are now thinking about way to optimize this, or we are finding collaborators to solve this problem together, because we now have 4,000 samples that we are really confident about that they have been well created. So that's the reason why now we can measure this accuracy of the LLM models. But in the end, I think this will lead to very interesting potential possibility that right now, today, I show you is that automatic data collection and the manual creation of the single cell and spatial data is quite powerful. But then at the end, as a perspective, I show that maybe we can make AI model can collect all this data for us automatically. And this would lead to the interesting concept called the self-growing database. And with this self-growing database and with the large language model that Dr. David has explained this morning, I think in the end, all the bioinformaticians might lose their job, but eventually we will have a really smart AI model that can tell us about all the details in biology. And eventually, I think this data integration effort across diverse contexts will lead us to the fully multi-scale GRN that we can consider it as the real entity of the human digital twin. So I'll end my talk here, and all this work that I presented today, there are a lot of manual efforts that has come up from our lab members, especially Dr. Junho Kang and Song-Young Kim has led the project, and many other students are all participating in making this pipeline running together. And there are many good international collaborators, and I'll take questions.